Okay, a bit of an informal video today, no editing to make my talking sound like it's sped up. I thought it'd be a good idea to take a look at this pandemic going on right now and talk about what's new that you guys may not have been keeping up with. I know we're all too busy to be reading mountains of scientific journals and articles, which, let's be honest, are boring to begin with. Okay, let's start with vaccinations. Things that are going a lot smoother than expected. You never know what's really going to happen with the new vaccine you develop, so to see this much success, I'm actually getting hopeful that we'll see a working vaccine sooner than later. On Twitter, I briefly mentioned about the success of the Russian vaccine through Phase 1 and Phase 2, which is called Sputnik V, by the way. I don't know if I'm pronouncing that right. So for new vaccine development, one of the most important things is to check if the subjects have proper responses to it, aka test to see if they've produced a sufficient level of antibodies. Because a vaccine is useless if no antibodies are produced, or only produced for a small percentage of individuals. Sputnik seems to give antibodies to 100% of the individuals for 76 participants. The study was published in The Lancet, so why don't we have a look? I'll just read the abstract and method for you guys since you probably don't care about the rest of the article anyway. Aren't scientists just so nice? They have a portion at the top for casual readers in case they don't want to read all the sweat and tears they've poured into the study. We developed a heterologous COVID-19 vaccine consisting of two components, a recombinant adenovirus type 26, RAD26 vector, and a recombinant adenovirus type 5, RAD5 vector, both carrying the gene for severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, spike glycoprotein. We aim to assess the safety and immunogenicity of two formulations, frozen and lyophilized, of this vaccine. We did two open non-randomized phase 1/2 studies at two hospitals in Russia. We enrolled healthy adult volunteers, men and women aged 18 to 60 years to both studies. In phase 1 of each study, we administered intramuscularly on day 0 either one dose of RAD26S or one dose of RAD5S and assessed the safety of the two components for 28 days. In phase 2 of the study, which began no earlier than 5 days after phase 1 vaccination, we administered intramuscularly a prime boost vaccination with RAD26S given on day 0 and RAD5S on day 21. Primary outcome measures were antigen-specific humoral immunity, SARS-CoV-2 specific antibodies measured by ELISA on days 0, 14, 21, 28, and 42, and safety, number of participants with adverse events monitored throughout the study. Secondary outcome measures were antigen-specific cellular immunity, T-cell responses and interferon gamma concentration, and change in neutralizing antibodies detected with SARS-CoV-2 neutralization assay. These trials were registered with clinicaltrials.gov, blah, blah, blah. That's funny because I actually looked in clinicaltrials.gov and I actually couldn't find it. I was able to find it here, though. Findings. Between June 28 and August 3rd, we enrolled 76 participants to the two studies, 38 in each. In each study, 9 volunteers received RAD26S in phase 1, 9 received RAD5S in phase 1, and 20 received RAD26S and RAD5S in phase 2. Both vaccine formulations were safe and well tolerated. The most common adverse events were pain at injection site, hypothermia, headache, asthenia, and muscle and joint pain. Most adverse events were mild and no serious adverse events were detected. All participants produced antibodies to SARS-CoV-2 glycoprotein. Looks very promising so far. I'm impressed. Remember when Russia said they would create a safe and working vaccine for distribution by October? I made fun of it saying it's not happening. Don't get me wrong, I still don't think it's happening, especially the distribution part, but perhaps we'll see something before the end of the year. If things go smoothly from here, which has a pretty good likelihood, we may see a vaccine so much sooner than anticipated. And the thing is, Russia isn't the only one who has been seeing success. We've also seen it in Sweden, who has passed phase 1 and phase 2 with their new vaccine, NVX-CoV2373. We initiated a randomized placebo-controlled phase 1-2 trial to evaluate the safety and immunogenicity of our SARS-CoV-2 vaccine in 5 micrograms and 25 microgram doses with or without matrix M1 adjuvant, and with observers unaware of trial group assignments, in 131 healthy adults. In phase 1, vaccination comprised 2 intramuscular injections, 21 days apart. The primary outcomes were reactogenicity, laboratory values, serum chemistry and hematology according to Food and Drug Administration toxicity scoring to assess safety, and IgG anti-spike protein response in ELISA units. Secondary outcomes included unsolicited adverse events, wild-type virus neutralization, microneutralization assay, and T-cell responses, cytokine staining. IgG and microneutralization assays results were compared with 32 IgG and 29 neutralization convalescent serum samples from patients with COVID-19, most of whom were symptomatic. We performed a primary analysis at day 35. After randomization, 83 patients were assigned to receive vaccine with adjuvant and 25 without adjuvant, and 23 participants were assigned to receive placebo. No serious adverse events were noted. Reactogenicity was absent or mild in the majority of patients, more common with adjuvant and of short duration, meaning less than or equal to 2 days. One participant had mild fever that lasted 1 day. Unsolicited adverse events were mild in most participants. There were no severe adverse events. The addition of adjuvant resulted in enhanced immune responses, was antigen dose sparing, and induced a T helper 1 response. The 2-dose 5 microgram adjuvanted regimen included geometric mean anti-spike IgG and neutralization responses that exceeded geometric mean responses in convalescent serum from mostly symptomatic COVID-19 patients. So yeah, overall it seems good things are happening. I wouldn't get too excited though, because don't forget there's still that nasty phase 3 trial up ahead. And then after that, who knows how long the vaccine will even last for. Interesting, isn't it, that this may be added to the list of recommended vaccines for children. Anti-vaxxers are going to be so mad. Anyway, in other news, it appears that we have some updated information on hydroxychloroquine. If you've been following my channel for a while, you know that there's a lot of conspiracy theorists who are claiming hydroxychloroquine is a magical cure for the Dark Lord. Thanks, Trump, for contributing to the spread of that false information, by the way. Anyway, there are a lot of variations to this claim. The video I made a while back with a bunch of doctors spreading pseudoscience claimed that this drug is effective at low doses. They claimed that it didn't work elsewhere because they give too high of a dose, and I actually broke this down a little bit in my video. Anyway, it turns out that a new paper, well, I say new, but it's been out ever since the end of August, claims that low-dose hydroxychloroquine may actually be effective. So yeah, usually I keep up with news on this pandemic through articles reported in newsletters, but as a result I don't receive information on new research that, shall I say, isn't very groundbreaking. So I didn't know about this until recently when I received a request to talk about this. Here's a video by Dr. John Campbell. Don't get me wrong, he's not a conspiracy theorist or anything, but he made a video that garnered a lot of attention talking about hydroxychloroquine. Let's have a look.
Now, I've had so many requests to look again at hydroxychloroquine, and that's exactly what I've done. And I've found a study from Belgium that gives fairly strong evidence, in fact I would say quite strong evidence, that low-dose hydroxychloroquine is an effective treatment. And the key seems to be that they used a much lower dose than the other studies. You know, there's quite a few things in this pandemic and you just can't work out why people didn't seem to realise things or get things so wrong. So this is the study from Belgium. Low-dose hydroxychloroquine therapy, right? So that's the study there. Now let's notice it's an observational study, so that's not ideal, as we know. We like randomised, double-blind, controlled trials when we can, but an observational study of 8,075 patients is a significantly large study. This is potentially good stuff, and it's fully published. It was published on the 24th of August, International Journal of Antimicrobial Agents. So the background to this study is hydroxychloroquine has been largely used and investigated as a therapy for COVID-19 at a total dose usually ranging from 2,400 milligrams to 9,600 milligrams. Right, in Belgium, off-label use, that means it's not officially used for that purpose, but it was, of low-dose hydroxychloroquine. And they gave us a total of 2,400 milligrams over the 5 days. Now, in the groups they came up with, they were, first of all, there was hydroxychloroquine alone with supportive care. When I say alone, they didn't give it with anything else. It was just hydroxychloroquine and all the usual care that the patients got, of course. All the usual care as well as. So the hydroxychloroquine was as well as the usual care. And then the second group, so that was the first group. And if you like, the control group, they just got supportive care only. They didn't get the hydroxychloroquine. There was 4,500 patients in that. Good number, very good number in fact. And in that group, 17% died during the hospital stay. In the group where there was no hydroxychloroquine, 27% died during the hospital stay. And for these numbers, that is a very significant result. So there we go. This study in Belgium is a strong indicator of efficacy for correctly dosed hydroxychloroquine.
Okay, so he does mention in the video that this is an observational study, which is good. It's something you can't really overlook, and it's in the title too. In case you guys don't know, an observational study is very different than what we normally want to do for clinical trials, which are double-blinded randomized studies. So also, just in case you don't know what they are, an observational study usually involves looking back at a situation after it has happened to try to determine a pattern. Meanwhile, the optimal type of study, a double-blinded study, involves sorting patients into two groups and then specifically testing your treatment against a placebo. So let's take a look at things that are missing in an observational study. First, things are in hindsight. It's much easier to be biased and cherry-pick patterns you're looking for in order to fit your hypothesis. Second, there's no placebo group. In this study, they looked at people who received regular ICU care plus hydroxychloroquine vs people who were just on the ICU care. The latter is not a proper control group because it doesn't take into account placebo. Third, the patients are not blinded. They know what they're receiving in terms of treatment. Fourth, external factors are not controlled for, which they normally would be in a randomized double-blind trial. Of course, you can still sort your data by age group, sex, medical history, clinical features, and have a well-defined inclusion/exclusion criteria, which is what the study did to their credit, but that overall would still be less controlled compared to a double-blinded study, which will have all of that plus more. In general, an observational study is weak compared to a randomized double-blinded study, so those studies by themselves is usually not sufficient enough to prove anything. Now, does that mean that observational studies in general are worthless? Absolutely not, but the fact that they're much more susceptible to factors I mentioned above, plus other factors such as selection bias, makes them a little bit more dubious. But anyway, what do I think of the study itself? Well, even the authors admit that some things could have been done better. For example, the evaluation did not have the primary purpose of determining hydroxychloroquine efficacy, and that the doses were not systematically checked for, both of which, by the way, would not be an issue in double-blinded randomized studies. Now, the authors are not some kind of conspiracy theorists or anything. They even stated, although observational studies, even of large scales, do not provide final conclusions on treatment efficacy, the results are important to consider to guide clinical trials. And then later they say evidence of an antiviral activity on hydroxychloroquine on Lord Voldemort seems increasingly inconsistent. Which, yes, I agree with. This study definitely does not determine anything by itself and will need further clinical trials. Double-blinded studies specifically. Except that kind of already has been done and results were not promising. This is the reason why newsletters and outlets are not reporting this article. It doesn't really give us any new information, and observational studies are just sometimes not very useful anyway. Regardless, there are so many other candidates right now, so I personally don't think there's much merit in hanging on to hydroxychloroquine. That being said, I do think the authors did an incredible job. They have a massive sample size, which increases the robustness of the study, assuming there aren't any other errors in it. Dr. John Campbell also mentioned that this study is different due to the dose it gives, which I agree with. At the end of the day, I think he and I differ in opinion on how much an observational study weighs, so I personally am going to hold off on making any conclusions about low-dose hydroxychloroquine. Anyway, that's the end of the video today, guys. Thank you to everyone who watched until this point, and thank you to my patrons, Fireshar, Liam, Alan Morton, and JN. See you later.